So we're going to take just a few moments, as you can see from the title of the slide, and talk a little bit about chemistry of microbes. Now, yes, I'm very aware this is microbiology and not chemistry. However, when we discuss microorganisms and we get into the next unit and we look at the structure of those microorganisms, you do have to have a very basic understanding of each of the 4 macromolecules. Now, I'm not talking about a lot. Literally, it's more of definitions that you need to know. So just a little bit of information about the 4: carbohydrates, lipids, proteins, and nucleic acids, so that we can build on that knowledge when we get to the next unit. So let's start with the most abundant out of the 4 macromolecules, and let's look first at your carbohydrates. Now, your carbohydrates are your sugars, and the carbohydrates in their simplest form will have that chemical formula of CH2O. Okay, and then how many of those that you build together to form that polysaccharide, that carbohydrate, is indicated by that number N there, and that N is those numbers of that piece that you put together. Now, when you just have one, it's known as a monosaccharide, okay? Or if you put 2 of those CH2O units together, then it's a disaccharide. So the building blocks of carbohydrates are going to be those simple sugars: the glucose, the dextrose, and the sucrose. Now, we take those simple sugars and we combine them together in long chains, and we call it a polysaccharide. And polysaccharides are very important for us to understand because, first of all, since they're sugars, they are going to serve as a nutrient or energy source. But don't forget, they're also going to be, you combine those sugars with pieces of proteins, etc., and they form structural units. So you're going to really have to understand that structural unit of the cell wall in the next unit. So you need to understand the carbohydrates and the polysaccharides just as that component, okay? Now, lipids, that's our next, that's number 2 out of the 4 macromolecules, and lipids are the fats, okay? And as a fat, they are actually going to be a triglyceride, and so their function is going to be of storage. Now, when we talk about a triglyceride, it's going to be having a glycerol with 3 fatty acids. There are 2 forms of triglyceride, and for those of you who are into nutrition and stuff, you're very aware of this already, but it's got saturated and unsaturated fats, okay? So when it is a triglyceride which has saturated fatty acids, they're going to be solid. They're going to be solid at room temperature like your butters. Whereas your unsaturated fatty acids, they are going to be the oils. They're going to be liquid at room temperatures. And one thing to recognize is with bacteria and cells, they generally keep their fats in a liquid droplet form. Okay. Now, let's talk just a little bit about 3 ways that those triglycerides can be modified and that we're going to be able to use them for structural components in our cells. And the first is going to be a phospholipid. Now, getting into microbiology, you had to have your prereqs, and I know you've all heard of the phospholipid bilayer or the cell membrane. Okay, so remember, phospholipids, they've got 2 components. Okay, they have a head which is hydrophilic, water loving. Okay, and then they have their tails which are hydrophobic or water fearing. So you can have your phospholipid, and it can be in a single layer or it can be that bilayer, which is the double layer. And the bilayer is the one we mostly refer to. And what you can see here from the diagram is because the heads are water loving, if you put the phospholipids together, the heads go to the outside near the water, and then they force the tails, which are water fearing, the hydrophobic pieces, to the inside. Okay, so just remember phospholipids is part of the lipid group, and it's used for the cell membranes. 2 other types of lipids out there are the steroids and the waxes. Now, steroids are going to be rigid, they're going to be complex lipids out there, but they are going to be forming cholesterols, and cholesterols are going to be needed for cell components and cell membranes when we get to the next unit. And then there's the waxes. Now, the waxes, again, are long chain lipids. They're going to have some alcohol embedded in with them, but they're a saturated fatty acid. And remember what we said on the first slide with the lipids is that if it's saturated, that means it's going to be solid. So waxes, as you know, were solid at room temperature, and this is going to be a real important component when we get to the particular mycobacterium that cause such things as tuberculosis and leprosy. And it's because of that wax being embedded in their membrane that's going to make them so potent as a disease pathogen. Okay, now let's talk just a little bit about proteins. Now, proteins are an predominant organic molecule in cells. So you hear a lot about the proteins, okay? Now they have a components that when you take a protein, and remember it's a macromolecule, which means it has to have all these subunits and we have to build it up. But one of the things to understand about proteins is that its shape is what makes it functional. So we got to talk a little bit about how we get the protein to its functional shape. And it has building blocks, and those building blocks are known as amino acids. And there are 20 different amino acids, and how we put those together will then form that functional protein. So let's look a little bit at the 4 steps to building a protein. The first one is, as we said, you got 20 different amino acids. Think of those as 20 different colored beads, okay? And so you have a whole bag of them, and you just get to pick them and put them in any order that you want, okay? That is the primary structure, the way that you put those different alanines and glycines, etc., in order. Those amino acids, the order they go in, that's the primary shape. All right, now you take all those beads and you're going to take them and you're going to string them and put them in an order, okay, and you're going to then fold, and that's your secondary. And then you take it and you twist and you put that whole bead into, you're going to continue to fold it, if you will. Think of just putting it into the smallest, most compact little ball that you can do. That's the tertiary, okay? So you keep adding, and every time you add additional folds and stuff, you're adding another bond. So then you've taken that beads, you've strung them together, you've folded them up, and you've squeezed them into the tightest little ball that you can do, the tertiary structure. You put several of those together. Then when you put several of those little string beads together, that is the quaternary, or the 4th structure. And when you start putting those all together, that builds a large complex protein. Now, why are proteins so predominant in the cells? Because they're used for there's so many functions. Let's talk about a couple. One is just enzymes. You need proteins. Enzymes, all enzymes are proteins, but not the opposite. Not all proteins are enzymes, okay? Enzymes are the catalyst for so many chemical reactions within the cells, and they're very important part to every cell being able to survive. Another use of proteins is their structural uses in the cell, and they also were things like antibodies, okay? When we get to the antibodies, I believe that's in chapters, or in Unit 6, you'll learn a little bit more about how they are made up out of proteins, okay? So you do need to understand that proteins are complex. You build them from amino acids, and then you got to fold and bond those amino acids to form these complex proteins, and that proteins are dependent upon their shape to be functional. And why all that's going to be important is because when we get to Unit 3 and we talk about how do we control bacterial growth, we're going to have all kinds of various agents that we're going to use, and the whole goal of many of those agents is to denature a protein. So we'll use heat, acid, alcohols, disinfectants, things along those lines, because if you can denature a protein, it means you've broken its shape down, and if you've broken its shape down, then it no longer functions. So think about a key going into the door. The shape of the key determines if it functions to open the door. If you bend that key or break that key and you alter that shape, that key no longer works. Same principle with a protein. So many things we do, and when we get to the next couple of units and we're starting to talk about how these microorganisms look in the next unit, and then in the 3rd unit we're going to talk about, hey, we can exploit that fact. If we know what proteins are used to help make that cell functional, if we break down that protein, then we can actually destroy the bacteria or the virus or whatever that microorganism is. The last component that we're going to talk about, the last of the macromolecules, are the nucleic acids. And a lot of times students, as soon as they hear of nucleic acids, they think genetic material, and that is obviously correct. DNA and RNA are both genetic material, and they belong to the macromolecule group of nucleic acids. But the one that sometimes students forget is ATP. ATP is actually also classified as a nucleic acid because of the fact that it does have that phosphorus group, which is a requirement, and it's going to have one of the nitrogenous bases. So in DNA, the nitrogenous bases, you may remember, are adenine, thymine, guanine, and cytosine, the A, T, C, and G. And then in RNA, it's adenine, uracil, guanine, and cysteine. So it's A, U, G, and C. Okay, so just again, and then when you talk about ATP, it's adenine, and it's got the triphosphate group on it. So it too has that structure. And again, we use the nucleic acids for energy and for genetic material. So, as mentioned, just a very quick overview, just trying to give you definitions again of the 4 macromolecules, how they're used by the cells, so that we can build on that knowledge in the next couple of units.